Plant callus is a mass of unorganized parenchyma cells derived from plant tissue for use in biological research and biotechnology. In plant biology, callus cells are those cells that cover a plant wound. Callus formation is induced from plant tissues after surface sterilization and plating onto in vitro tissue culture medium. Plant growth regulators, such as auxins, cytokinins, and gibberellins, are supplemented into the medium to initiate callus formation or somatic embryogenesis. Callus initiation has been described for many plant taxonomic divisions. Parenchyma, Anthoceratophyta, Bryophyta, Lycopodiophyta, Pteridophyta, Cecadophyta, Ginkgophyta, Pinophyta, Gnatophyta, Magnoliophyta. Callus induction and tissue culture. A callus cell culture is usually sustained on gel medium. Callus induction medium consists of agar and a mixture of macronutrients and micronutrients for the given cell type. There are several types of basal salt mixtures used in plant tissue culture, but most notably modified murashige and skug medium, White's medium, and woody plant medium. Vitamins are also provided to enhance growth such as Gamborg B5 vitamins. For plant cells, enrichment with nitrogen, phosphorus, and potassium is especially important. Morphology. Plant callus is usually derived from somatic tissues. The tissues used to initiate callus formation depends on plant species and which tissues are available for explant culture. The cells that give rise to callus and somatic embryos usually undergo rapid division or are partially undifferentiated, such as meristematic tissue. In alfalfa, Medicago truncatula, however, callus and somatic embryos are derived from mesophyll cells that undergo differentiation. Plant hormones are used to initiate callus growth. Specific auxin to cytokinin ratios in plant tissue culture medium give rise to an unorganized growing and dividing mass of callus cells. Callus cultures are often broadly classified as being either compact or friable. Friable calluses fall apart easily, and can be used to generate cell suspension cultures. Callus can directly undergo direct organogenesis and or embryogenesis, where the cells will form an entirely new plant. Callus cells deaths. Callus can brown and die during culture, but the causes for callus browning are not well understood. In Jatrophaecoccus callus cells, small organized callus cells became disorganized and varied in size after browning occurred. Browning has also been associated with oxidation and phenolic compounds in both explant tissues and explant secretions. In rice, presumably, a condition which is favorable for Scitella callus induction induces necrosis too. Uses. Callus cells are not necessarily genetically homogeneous because a callus is often made from structural tissue, not individual cells. Nevertheless, callus cells are often considered similar enough for standard scientific analysis to be performed as if on a single subject. For example, an experiment may have half a callus undergo a treatment as the experimental group, while the other half undergoes a similar but non-active treatment as the control group. Plant calli can differentiate into a whole plant, a process called regeneration, through addition of plant hormones in culture medium. This ability is known as totipotency. Regeneration of a whole plant from a single cell allows researchers to recover whole plants that have a copy of the transgene in every cell. Regeneration of a whole plant that has some genetically transformed cells and some untransformed cells is called a chimera. In general, chimeras are not useful for genetic research or agricultural applications. Genes can be inserted into callus cells using ballistic bombardment, also known as a gene gun, or Agrobacterium tumefaciens. Cells that receive the gene of interest can then be recovered into whole plants using a combination of plant hormones. The whole plants that are recovered can be used to experimentally determine gene function, or to enhance crop plant traits for modern agriculture. Callus is of particular use in micropropagation, where it can be used to grow genetically identical copies of plants with desirable characteristics. History. Henri-Louis de Hamel du Monceau investigated wound healing responses in elm trees, and was the first to report formation of callus on live plants. In 1908, E. F. Simon was able to induce callus from poplar stems that also produced roots and buds. The first reports of Callus induction in vitro came from 3 independent researchers in 1939. P. White induced Callus derived from tumor-developing precambial tissues of hybrid Nicotiana glauca that did not require hormone supplementation. Gawther and Novacourt were able to maintain Callus cultures of carrot using OIN hormone additions. See also, embryo rescue, somatic embryogenesis, chimera, hyperhidricity. References